So, Dr. Shulman, congratulations, a really nice paper. Can you tell us just a brief summary of what the main features were?
Sure thing, and thanks for inviting me for this conversation. So what we set out to do is examine the genetics of amyloid pathology deposition in the brain. Most of the focus in Alzheimer's disease genetics has been to study clinically diagnosed cases or control subjects. One of the problems therein is that there's a lot of pathology for amyloid we now recognize in individuals who haven't been diagnosed with Alzheimer's disease yet. So this particular study actually uses directly measured amyloid pathology as an outcome trait in order to do genetic analyses. And we had two specific questions. One, we asked whether known, published, and validated Alzheimer's disease susceptibility genes actually affect the deposition of amyloid pathology in a large autopsy cohort. And two, whether using that outcome measure, directly measured pathology, allows us to discover new genes that we didn't know about. And the basic findings of the study were on the first question, we identified that 3 of the 9 or so discovered Alzheimer's disease susceptibility genes to date are associated with directly measured amyloid pathology. Those are the complement receptor 1, the ABCA7 transporter gene, and the CD2AP gene. Those 3 genes came from genome-wide association studies that have been published in the last couple of years. And then in our genome-wide association study, which was our effort to find novel genes associated with amyloid pathology, the only significant finding from that study was a locus we already knew about, that of ApoE, and we additionally found several interesting new genes with suggestive associations, and one of those is an association signal very close to the amyloid precursor protein gene, which of course is a gene that's mutated in rare familial forms of Alzheimer's disease.
So the innovation here is you're looking at autopsy and you're using a direct neuropath marker.
That's absolutely right, and I want to point out that this type of study would not be possible without large, well-collected autopsy cases, and in this case we had the advantage of using cohorts that are led by David Bennett at Rush University, and those are the Religious Orders Study and Rush Memory and Aging Project, and those studies have collected nearly 1,000 autopsy cases over the last nearly 20 years. Individuals are without dementia at the time of entry into the study. The study actually, as I mentioned, began 20 years ago. People are followed prospectively, and then when they die, they have consented to brain donation. So we have an opportunity to kind of see them develop dementia, have the availability of clinical examinations within the year before they died, and then an opportunity to look at their brains and ask what the pathology was. And of course now with genomics technology that have really been led by another mentor of mine, Phil Dieger at Harvard Medical School, we have access to genome-wide genotyping for these individuals, so we can relate genetic changes to directly measured amyloid pathology in the brain, and of course to their clinical status proximate to death.
So coming on to that, my understanding is that the new association, the new SNP, which is close to APP, didn't appear in the large clinically based cohort genome-wide association studies to the US. Did that surprise you, and how do you interpret that?
Yeah, I think that's of course quite intriguing, and it needs to be followed up with further studies to understand the true explanation for that. One thing I want to first point out is that, as I mentioned, the association that we see near APP doesn't reach the strict significance criteria that our genetics field typically uses, although it is strongly suggestive. What that means is that in order to know that it's in fact true, additional replication will be required in independent studies, and that's something that we're actively pursuing. And we, in fact, are presenting the work to encourage others to also try and replicate the finding. And another caveat that I want to point out is while this association peak is close to the amyloid precursor protein gene, it's not a coding variant. It doesn't change the protein sequence of the gene, and it's possible that its primary effect would actually be on another nearby gene. But APP is clearly the most intriguing and compelling candidate gene nearby this signal. So to specifically address your point, though, I want to make the important distinction that what we've done is different from the genome-wide association studies that are being done in Alzheimer's disease. Instead of comparing cases and controls, individuals with Alzheimer's dementia to those who are non-demented and age-matched controls, our population cohort has a range of clinical diagnoses at the time of death, including some individuals with no cognitive impairment, mild cognitive impairment, or in fact dementia. But the outcome measure is irrespective of those clinical diagnoses and is looking at quantitative amyloid pathology, which is in fact relatively normally distributed within that population. So it's possible that we would find an association to amyloid pathology that would not be seen in an Alzheimer's disease case control analysis, because we recognize that amongst controls that are older in the population, there will be a substantial amount of amyloid pathology there. So it could be something that would be missed in an Alzheimer's disease case control analysis because of that potential for latent confounding. It's possible that larger sample sizes in a case control analysis would discover such an association. In our smaller data set with about 700 or so autopsies, when we look at Alzheimer's disease cases compared to Alzheimer's disease controls, we see no association at this SNP to Alzheimer's disease diagnosis. However, when we do a further analysis and restrict consideration to those individuals with clinically diagnosed disease and pathological confirmation, both on the case and control side, in other words, we remove those control individuals who actually have Alzheimer's disease pathology at autopsy, we in fact do see the association. So it suggests that the confounding in the case control analysis that I described is in fact present.
I mean, this is a great resource you've got here. So I guess you're looking at other neuropathological markers like tau deposition or synuclein?
Yeah, that's absolutely one of the next steps. And one of the values of the Rush University cohorts, the Early Disorder Study and Rush Memory and Aging Project, is the comprehensive neuropathological evaluation led by the neuropathology team there. And Julie Schneider, who's the lead neuropathologist on the studies, is one of my co-authors and collaborators. So indeed, we have information about neurofibrillary tangle burden. We have information about the presence of synuclein pathology, and we know that all of these pathologies, including cerebrovascular pathology, are important contributors to dementia, and many pathologies besides the pathology of Alzheimer's disease that we recognize may contribute to the phenotype of dementia that occurs in populations. So we are looking at all those pathologies.
Well, again, congratulations, Josh. Very nice study, and thanks for sharing it with us.
Thank you very much for your interest.